All right, well, it's my pleasure to introduce Stephanie Koontz. Stephanie's been working at Archbold for about 9 years. She's multi-talented in the field and with data and modeling. She came to us with a master's degree from Maryland and has also worked at the Avon Park Air Force Range. And today she's going to talk about a long-term demographic project comparing wild populations to translocations, which is introductions and augmentations in the very rare plant Dicerandra christmanti. Stephanie?
Thank you, Eric. I'd like to thank everyone for coming today. I'm excited to speak with you about how myself and my co-authors were inspired by nature and how we used wild populations to inform rare species translocations and evaluate their success. Maybe. It was working a minute ago. Let's see. Sorry. There it goes. Okay, so preserving biodiversity is critical in the face of an ever-changing world. Protecting our natural areas is the most effective measure we can take in our efforts to conserve this biodiversity, or that remains. However, even if we have protected areas, not all species, especially plants, are able to disperse to these protected places. Therefore, over the last few decades, translocations have been viewed as a vital tool in our efforts to preserve our biodiversity. So before I go any further today, I wanted to review a couple of definitions that I'm going to use frequently throughout this talk. We define translocations here as a human-mediated movement of a species from one location to another. These can include introductions, which moves a species to a previously unoccupied site. We can also have a reintroduction in which the species was historically known to occur there, but no longer does. We can also have an augmentation, which is when you add propagules to an existing population, to just kind of help bolster the population size or help keep it from going extinct. Today, I'm going to focus mostly on an introduction and an augmentation that we did. Whoops, okay. The goal of a translocation is to create viable populations. Several metrics have been identified in the literature for how to measure translocation success. These metrics include flowering and fruiting to produce seeds, but then those seeds must also be able to germinate, survive, and produce viable seeds for the next successive generation. Populations also need to be able to disperse and expand into new appropriate habitats. And overall, we expect that translocated populations should eventually be able to mimic natural processes as we observe in wild populations. So Archbold and the work we do mostly takes place here on the Lake Wales Ridge. Central Florida is unique in that it is traversed by ancient sand dune ridges. These relics of higher sea levels are sand dune beaches that were left behind as the ocean receded over 2 million years ago, but they're now home to many of the endemic species found in Florida and in Florida scrub. The Lake Wales Ridge is the oldest and longest ridge. It's about 150 kilometers long, and Archbold Biological Station is situated at the southern end of this ridge. Florida scrub is a shrub-dominated landscape. Historically, lightning strike fires managed the land at intervals ranging from 3 to 60 years, depending on the habitat. Many of the species found here have adapted to frequent fire and depend on these fires to maintain suitable habitat. The Florida scrub has high levels of endemism, with many species found only on these sandy ridges and nowhere else in the world. In fact, these ridges make up about 1% of Florida's landmass, but they account for about 40% of the federally listed species found in our state. One of these the species I'm going to talk to you today about is in the genus Dicerandra. It's also one of the rarest in the Southeast. The Dicerandras have a very unique and diverse history along these ridges with about 10 accepted species, and this is their range here in the map. Most of these species are limited to only a few counties, and there's very little overlap among these species ranges. And the one I'm going to speak to you today is at the southern end of all these ridges, Dicerandra christmanti, found only in Highlands County, along with Dicerandra frutescens, the hot pink highlight there in the middle center of the state. Whoops, too far. Dicerandra christmanti is a G1 critically impaired species endemic to Florida, and it's found only on yellow sand soils of Florida scrub, growing in sandy openings within the shrub cover. These openings are known as gaps, and they're not very big. They're only a few square meters in area, but they're home to many of the endemic species that we work on, including Dicerandra christmanti. Dicerandra christmanti is known from only a few sites, one of which is protected, the Flamingo Villas, at the Flamingo Villas, part of the Lake Wales Ridge National Wildlife Refuge. And on this map on the right, you can see the property there in the bottom part of the map, and the populations are there highlighted in pink. There's a few unprotected wild populations just north of this property that are currently not under conservation. It's been recommended by the U.S. Fish and Wildlife Recovery Plan, which exists for all listed species, that translocations need to be used to aid in the recovery of this species. So our program has both augmented the Flamingo Villas population there on the southern end in 2010, and we also introduced a new population in 2012 to Carter Creek, also part of the National Wildlife Refuge System and just north of these wild populations. So I'm going to talk a little bit about the wild sites first. We've collected demographic data at wild populations at Flamingo Villas since 1994 and 1999 is when we started some of our long-term transects. As you can see, we have lots of observations, over 5,700 observations of individual plants. And we monitor these in roadsides and also in open sandy gaps. And so I kind of did an aerial so you could see what we're working with. And on the left photo, we have, you can see the little bits of white, and those are our very small gaps and where many of these endemics occur. We also have sandy roads that are maintained to be open as fire lane access for land management purposes. And as I mentioned, we did translocations with only a few hundred plants, but they quickly expanded into thousands of observations. We augmented the Flamingo Villa population into previously unoccupied gaps in 2010. We added 200 plants, and by 2018, we had over 6,000 observations. Similarly, we did an introduction in 2012 where we added just over 200 plants to unoccupied gaps at Carter Creek National Wildlife Refuge. And within just a few years, we already have over 5,000 observations. Our methods for sampling these populations are the same so that we can compare our data. We visit every single marked plant and gap that we monitor annually in October. This is when the plant is flowering. We monitor survival. We record the life history stage, whether it's a new seedling, a vegetative plant, or a reproductive plant. And then we get size measures based on the total number of branches, but then also the total number of flowering branches. So our study sites, we refer to them as management treatments. This is because the ecological and human disturbance vary among them and therefore have implications on the demographics. For example, we have our natural undisturbed wild gaps that are closing an area due to lack of fire. Our roadside habitats, however, remain open in the human-created sandy fire lanes. Translocations were managed due to the design. By selecting only good quality habitats and using well cared for large propagules, and we initially provided supplementary irrigation, we possibly inflated population dynamics. By treating these differences as management treatments, they add explanation to possible differences in our observed populations. So we did some preliminary analyses, which are kind of the basis for this research. We compared some very simple vital rates among the 4 management treatments to see how populations were comparing to each other or how they were performing compared to wild gaps and disturbed roadsides. The vital rates that we explored included survival, growth, flowering, and recruitment. And based on what we observed, we questioned if these were pictures of success. We can look at survival, annual survival, in our top left graph. And we can see that we have our 4 different management treatments. We have our wild gaps in red, our roadsides in black, our introduction is in blue, and our augmentation is in green. And we see that for most years, annual survival was similar among these 4. The one exception is the decline in the blue. This is because a prescribed fire came through and killed the adult plants that existed. But we can see for those that survived and those that had subsequent recruitment, their annual survival was very comparable to the other three management treatments. We can also look at recruitment density, which is on the top right. This is the number of seedlings per meter squared. And in most years, our translocations with the augmentation and introduction are very similar to our wild sites and our wild roads, and in many cases actually out-recruited those sites. We can also, in the bottom left, look at the change in growth rates. And so this is the number of branches in 1 year compared to how many branches there were the next year. This linear black line, or this horizontal black line, kind of indicates no growth. So if you're above that line, you are growing in a positive, you're getting larger. If you're below that line, you're getting smaller. And so we see in most years our plants are getting, for the most part, larger, but we see that with those green and blue dots kind of much higher than those black and red dots. That means our translocations are growing that much larger than the other wild populations. Similarly, we can also look at the proportion of flowering branches, which is in the bottom right. And so this is similar to our relative growth. It's out of all the number of branches that we have, what percentage of them were flowering. And we see a little bit of a pattern here again, mainly in the introduction and initially in the augmentation, to a higher proportion of those branches that each plant was producing tended to be producing flowers and therefore seeds that can be building up a seed bank. But even with all of these positive trends that we're seeing in these very simple exploratory analyses, I wasn't convinced. I had watched these populations flourish and transition along the rises and falls, and my concern was translocations don't have the years of demographic inertia to push through the lows. We could be witnessing what's known as a time lag response, in which recruitment conditions have been phenomenal, but long-term reproductive efforts may be less than par. And with the decline of both translocations in recent censuses, at what point would be too late to intervene to stabilize the conditions? So this led us to a couple different questions and hypotheses. First, we wanted to know what the dynamics of Dicerandra christmantii are. We have not done any modeling of this species to understand how even wild populations respond to environmental conditions. So our hypotheses, we assumed that wild gaps would be consistent annually, a little more stable year to year, but overall have a general decline, and this is due to most likely due to fire suppression in the flamingo village habitat. We also expected that roads would be highly variable, acting as more of like a sink population. So you might have really good boom years and really bad bust years. As for the translocations, we didn't really know what to expect. That kind of led to our next question, in which we wanted to know how they compared. And our hope was that they'd be more similar to our wild gaps, be a little more stable over time, and possibly not even showing those declines that we expected in the wild populations. But our fear was that they might be more similar to the variable road conditions. It's been shown in other endemic gap species that when they occur in these human-modified habitats, such as sandy fire lanes, they tend to be more variable in their population dynamics. And then finally, if we're able to answer those first two, our biggest question was going to be, were these translocations a success? Initially, I think we can say yes, but long-term trajectories can be hard to predict. So there's kind of two parts to these results sections that I'm going to talk about. First, we're going to talk about some of the population models. This looks very similar to what the former graphs that I showed you, but they're predicting responses based on the data that we have. So what we did is we partnered with some collaborators at the University of Central Florida, and they ran these models for us. And we were able to compare the vital rates of survival, growth, reproduction, and recruitment among our 4 management treatments. So first, we're going to start off with survival, and I'm going to walk you through these graphs. Remember, these are predicted curves. They're based on our real data, but they're not showing the real data on this figure. And so what we have is we modeled yearlings and adults separately. We expected that first-year recruits to act differently than established adults based on the environmental conditions for that year. And we have the similar colors as we did in the previous graphs, to where we have our wild gaps are in black, our roads are in red, we have the augmentation in green, and the introduction in blue. And that's for both our yearlings on the left and our adults on the right. What we have on our x-axis, which is the lower horizontal line, is the total number of branches observed. And based on an individual plant size, we're able to predict its probability of survival, which is on our y-axis. So what both of these graphs show, that as you increase in size, you tend to have a higher probability of surviving. Larger plants are more established, able to get resources, and therefore more likely to survive. What we can observe, or if we compare among our management treatments, we see a similar trend in both, to where wild gaps and wild roads are actually very similar. The introduction has a high probability of surviving across all, or more so than all of the other individuals or management treatments, but then also the augmentation has much lower probability of survival, which was kind of shocking to us. If we look over on the right in the adults one, in the adults figure, we see that there tends to be at larger sizes a leveling out or even a decline in probability of survival. That's expected as plants get larger, they start to reach the end of their life, and so they might actually start to see a decline in survival because they've been around for a while. Next, if we look at growth, again, we have yearlings on our left and adults on our right. We still have the number of branch tips on our x-axis, and it's increasing in size. But this time we have the predicted number of branch tips in your second year. So we wanted to know, are plants growing or are they decreasing in size from year to year? So this dashed line that you have going kind of horizontal across the middle of each figure is again showing positive growth on the top side and negative growth on the lower side. We see that for yearlings, they are positively growing, and obviously the larger you are, you're producing larger plants in the next year. However, the introduction is again producing significantly larger propagules than any of the other three management treatments. If we look at our adults, we see that same trend with really large plants occurring in the introduction. And there does tend to be a general decrease in size for larger plants. Again, this is most likely due to plants reaching the end of their life, and they tend to start to get a little bit smaller before they truly become deceased and leave the population. Next, we're going to look at reproduction. Here, we're only going to look at adults. Yearlings are able to reproduce their first year, but it's not very common. We didn't have enough data to really produce reliable models. So this is only looking at adults. And what we have now are, on the left, we have your probability of reproduction. So based on your size, were you going to flower that year, yes or no? And that's a changing probability scheme that we have there. And what we can see looking just at this one first is that kick out of that red line for road populations is actually, they had to be larger to increase their probability of survival, or not survival, I'm sorry, of reproduction. So they had to be larger to even start to flower. We see somewhat similarities between the other three management treatments, with wild gaps being able to be smaller and having a higher probability of reproducing compared to our translocations. But our translocations are falling right in that between those two, and so they're pretty comparable. If we look at the figure on the right, this is now fecundity. So this is the number of flowering branches in relation to the total number of branches that you have. In this case, that horizontal dashed line going across is 100% of your branches are flowering in that relation. So as we see, as you increase your total number of branches, you're increasing the number of flowering branches. But what I want to point out is in the lower end of this graph, our black line of our wild gaps is on top, indicating that they have fewer branches, but more of those branches are flowering. But then that trend actually tends to shift for larger plants. That larger plants, more of their branches are flowering for both of our translocation populations than would be expected in both of our wild populations. And then finally, we have our recruitment. This is total number of new plants for all four of our management treatments from year to year. And we see that there's variability within our wild gaps and our wild roads annually, so there's good years and bad years. But we see that when we first initially put those both those translocations out, they had huge recruitment episodes that have only in the last couple of years started to decline to levels more comparable to that what we observed in wild populations. So before I go on too much further, I wanted to review what we learned from our basic population models. We saw more or less more than 50% survival for adult plants and for yearlings across all four management treatments. Survival was definitely highest in the introduction, but tended to be much lower in the augmentation. We saw mostly positive growth, except for in larger plants nearing the end of their life. And again, the plants tended to be larger in the introduction. Reproduction probability was delayed in road sites, and fecundity was highest in our translocations, only for larger plants. Both translocations also had strong initial recruitment pulses, and so we're seeing similar, if not stronger, population dynamics in these translocations compared to our wild sites. So this is kind of just the first step that we did, looking at how these vital rates respond. Our second step was doing what's called integral projection models. So it's one thing if you have a vital rate such as survival or growth that is doing really well, but we also want to make sure that individuals are transitioning through their life stages. It does us no good if a ton of seeds are produced but they stay dormant in the seed bank. It does us no good if we get lots of yearlings that are produced but then they all die and never make it to their adulthood. So what we have here in this figure is kind of a progression of what individuals can do at different life stages. And I'm not going to go through all of them today. I'm definitely going to highlight some of them. But what it shows is that we can have seeds remaining in the seed bank. And so that's our step one with that arrow kind of going back. But we can also have then seeds that germinate and become yearlings, and that's our step two. Yearlings can then also, if they survive, will become adults, which is our step four. And then adults have multiple choices. They will remain adults in the alive within the population, and if they do, they'll be hopefully producing seeds through flowering and contributing either to the seed bank through step 8, or they can actually go ahead and produce seeds that germinate that year, and that's yearlings, step 5 there. So there's a lot of different possibilities and a lot of different steps, and like I said, I'm not going to go through all of them. I'm just going to go through some of the bigger steps today. But ultimately, what this show can be turned into is what's known as a heat map. And so again, we have those numbers for each of the steps, and warmer colors indicate there's a lot of input. There's a high probability of that step going on. And then the cooler colors indicate less or no probability. And this also is good or is helpful in that it actually has a size component, but I'll show that in individual figures. So I'm going to break this down for everybody and kind of walk through it one stage transition at a time. So our first stage transition is looking at seeds that are dormant in the seed bank, and then a portion of them will germinate and turn into yearlings. This is step 2 in the top right part of our figure there, seeds that are germinating, becoming first-year plants. And what I want to point out here is that we have, kind of cooler colors for smaller plants here, but those cooler colors are similar across the 4 management treatments. We don't have more seeds turning into yearlings in one management treatment over the other. So they're kind of all doing equal contributions of seeds germinating every year and becoming yearlings. We also then have yearlings that can survive and turn into adults. And here we also have, like I mentioned, an increasing size component. So everything on the left are really small yearlings producing that size adult. And as you move to the right, you get larger and larger yearlings. And what we see is there tends to be a general trend of larger yearlings are producing subsequently larger adults, growing year to year. The one I do want to point, oh, it's not in there anymore. I did at one point have highlighted, if you notice in the first figure for wild gaps, we tend to have this decline, which initially was kind of concerning that we had large yearlings surviving but producing smaller adults, which didn't really make sense to us. So we kind of went back and looked at the data, and what we realized is because, again, these are predicted models based on the data that we're able to provide, we just didn't have a lot of yearlings producing larger adults. And so based on some of the population models and how they work, or the projection models and how they work, we just didn't have a lot of data. So while it looks like there's a lot going on there, it's just not representative of what's truly happening in these wild gaps. We tend to see a similar trend to where the larger yearlings will produce a larger adult. And then the other thing I wanted to point out is if we look at that introduction, they all tend to have this same upward exponential curve, but in the introduction, it tends to be more elevated. So yearlings in the introduction are producing even larger adults in the next year compared to the other 3 management treatments. And then next up, we have adults that are remaining adults, surviving year after year. And again, we can compare those across the 4 management treatments. But what I want to point out is in both of our wild populations, for the gaps in the roads, we tend to have these really long extended tails. But those are kind of lacking in our augmentation or introduction sites. And so that was also a question that we had, is we're not sure if this is actually a truth within the data. Are plants just not getting as large in the augmentation and the introduction as we observe in our wild populations? The other question is also, these are much younger populations that we have created. The 2010 one, at the time of this modeling, we only had 8 years of data, versus the others, we have almost 20 years of data. So maybe just not enough plants have made it to that larger size yet. And so just the data don't exist quite yet. So again, we want to keep monitoring these to see if eventually those tails will mimic more of the wild roads and the wild gaps, or if it's going to remain this way, then maybe is there a question of why the plant's not getting as large in these two populations. So next we have seed bank contributions. And these are seeds that are in the seed bank and remaining viable, ready to germinate at whatever cues them. And those are red squares. There are a lot of seeds in the seed bank sitting there waiting to become first-year yearlings. So we have a really healthy seed bank based on our models. And then if we look at just adults producing seeds, we also see that across all 4 of our management treatments, there are a lot of adults putting lots and lots of seeds. So we are pretty confident there's a pretty well-established seed bank within all 4 management treatments. So going back to our questions, we want to know if these translocations were a success. So starting with our first question, what are the populations of dynamics of Dicerandra christmanti? We saw really strong vital rates across all 4 management treatments. We saw plants transitioning through those stages, from the seed stage to the yearling stage and into adulthood and surviving as adults. We also saw a lot of seeds being produced. So there's a very healthy seed bank that exists out there. And what was really surprising to us is that our wild gaps and roads were more similar than we expected. They didn't have the variability that we have observed in other endemic gap specialists, where along those roadsides, they tend to have much more variable population dynamics. So then how do translocations compare to these wild sites? Well, we saw overall the augmentation tended to be similar to our wild gaps and wild roads. Survival would be about the biggest one that was of concern was that it was lower than observed for the other 3 management treatments. However, survival is still well over 40% within adults for this augmentation. We also saw intermediate fecundity for the augmentation compared to the wild gaps, but as we saw, there's a lot of seeds going into the seed bank even for this management treatment. And overall, for all of our metrics, the introduction tend to be more robust, if not comparable, to our wild gaps. And so ultimately, can we declare these translocations a success? And I think yes, we can, but only with continued proper habitat management. So kind of in conclusion, for our future directions, we are going to continue to monitor both wild and translocated sites. We want to see how population dynamics continue to progress, especially in our translocated sites compared to our wild populations. We're working to determine how management treatments, even within our translocations, aided in our success. We did a lot of selection of gaps and appropriate habitat, and we think that has a lot to do with our overall observed success, but how do we manage the land to keep these gaps suitable for our translocations? We also want to continue to look at the effects of habitat management. There hasn't been a lot of data collected on the effects of Dicerandra christmanti in response to prescribed fire, but I'm really excited that recently the land managers of Flamingo Village have been able to get fire into our management units and have actually burned some of our long-term demographic plots. So we are going to start getting post-fire recruitment and survival dynamics for these wild populations. So we'll see how fire truly affects their populations, which is something we didn't used to have data on. But ultimately, we are going to continue with additional translocations to continue to secure unprotected sites, but also safeguard protected sites against future climate change and other habitat management. And with that, I'd like to thank my co-authors for designing and working endless years on collecting this data. This is one of the long-term data sets that Eric has put together, and so there's been multiple research assistants that have worked on it. There's been hundreds of interns that have been out there collecting this data. And it's great to have such wonderful data to be able to show such strong trends. We also like to thank U.S. Fish and Wildlife Service for allowing us access to their land, also for funding some of this research, and the National Science Foundation for also providing funding. And with that, I will take some questions, who I think Research Assistant Scott Ward is going to help me with.
Hey, Steph.
Hey, Scott.
So I don't have any questions yet, but they might start ringing in if I encourage people to ask questions. There was a comment by Carly Althoff, and I think she may have missed the methods of augmentation. So I'm not sure if she means she missed that part of the presentation or not.
Yeah, I didn't go too much into the detail of what we did, but I'm happy to do that now. So for both of them, we went out and we got stem cuttings from wild populations. For the augmentation, we used cuttings from flamingo villas because we wanted to use that genetic material. For the introduction up at Carter Creek, we, sorry about that. For the introduction, we actually got material from those unprotected populations that were just north of flamingo villas. We grew them up in pots and also had some help through Bach Tower Gardens helped grow some of those up for us, and just kind of managed them and grew them in pots in the greenhouse, and then brought them out and outplanted them. We did provide supplemental irrigation for the augmentation for all of our plants. They got irrigation for the first several months, mostly because we outplanted that in August, and so we were kind of getting into our drier fall season, and we wanted to make sure they were well established. For the introduction, some of the plants got supplementary irrigation and some did not for the first few months again, but we found no significant differences in survival or in size based on that irrigation. Now, we also had some really good wet years right after we did that introduction, so I think in the future that might actually have a treatment effect if we had a more drier fall like we usually get.
Okay, great. There are a couple of questions that have come in since. Dwayne Coulterman is wondering, have you done any studies of the species pollination biology?
We have not. There has been some work on Dicerandra frutescens, which is the other Highlands County endemic. I'm not quite sure exactly what that says off the top of my head, but they do get lots of pollinators. They are also subject to getting nectar robbers because there are certain little pollinators that are able to, or flies, I guess specifically, are able to come in through the side of the corolla and actually bypass the nectar or get to the nectar without getting to the pollen. But I don't know how much that is affecting their overall output. When we go and collect seeds for augmentation, we tend to get, it's in the mint family, so it's able to produce a little nutlet, so 4 little seeds per capsule, and we tend to get, I think, an average of about just over 3 what we consider a viable seed per flower. So they seem to be doing pretty well, and like we saw from our modeling, we see there's predicted based on these models, there's a lot of seeds going into the seed bank. And we've seen that in really good recruitment years, we'll get hundreds of seedlings in these really small areas. So while pollination is extremely important, especially understanding how it's interacting, how the pollinators are interacting with the plant, but also how it can affect the success of an introduction or an augmentation. As of right now, they seem to be getting a pretty good diversity of floral visitors.
Okay, we have another question from Kathy. She's curious, how does the introduction, or how do the introductions, augmentations, and translocations affect the local ecology of other plants? Is there any competition for resources between these species?
Within these little gaps, you know, that they tend to just naturally not have the shrub cover and competition that you would find within the shrub matrix. And so they do tend to have really high levels of diversity within these open sandy areas. That being said, with some of how successful some of these introductions and augmentations have been, there could definitely be competition going on with some of the other endemics. We haven't looked at that specifically. but I guess it's just, there's a lot of these gaps kind of scattered across the landscape. And so while you hope one species might be doing well and one that maybe another will be doing well somewhere else, but they tend to have most of these species tend to have pretty deep rooted systems, and water seeps down so quickly. So the access for water can be an issue in dry times, but if it's an issue for one, it's going to be an issue for all. It might just be more recruitment space that these plants can get rather large. And so if they're kind of blanketing this area, you're taking away gap space for other species to germinate within. So I guess you could say that if they did too well, they might be kind of stifling other endemics that are in that area.
Okay. We have a question for Becky. She's wondering, how did you decide on sites for new introductions? Were these based on historical records or observations, or estimated based upon habitat type?
So the augmentation, we wanted to just augment that population because of it had a fire suppression, has some fire suppression going on, but where we put the plants, they had actually just done a recent prescribed fire. So we were able to put plants out into gaps that had recently burned. So we wanted to see if that had a positive effect, and so far we think it definitely has. For the introduction, that was actually funding for U.S. Fish and Wildlife Service to create a new population. And you know, when we look at historical ranges, it's so hard to know what's historical, and you know, just based on nobody ever saw it there, does that mean it didn't occur? Now, Carter Creek is technically outside the range by only a few kilometers based on where those other unprotected wild populations are, but it is on the same yellow sand ridge that is connected all the way through those unprotected populations and down to Carter Creek, or sorry, down to Flamingo Villas. So Carter Creek was selected as it's one of the few national wildlife refuges that has the appropriate habitat type, but is also being actively managed as a Florida scrub sandhill ecosystem. So we knew if we put them out there, they would be protected and well managed. So some of it had to do more with just the habitat, and if we're going to put time and effort and money into this, we want to know it's going to be cared for and be a success.
Okay, good answer. The next question we have from Caroline Lanker is, so this study concerns one species. Do you plan to apply the results to other species of concern?
I mean, it leads us as, it provides us the basis of, we've seen this in one gap endemic, that maybe we can expect to see similar trends in others. However, I would still say that we would need to have some sort of data to be able to compare if we were doing translocations of other species, just because the life history of different gap endemics are so different that I think it would be presumptuous to kind of assume that what we saw with Dicerandra would work with another non-Dicerandra species. But I think we do understand in Florida scrub, though, that most of our endemics do need some sort of fire, and that most of them do respond positively to fire. So I think that there are some assumptions that you can get out of this study that would maybe help design an augmentation or introduction for another scrub endemic, but I think you'd still need to look more specifically or know something about the population dynamics of that species before you would start designing a translocation for that one.
Right. Okay. Next question is from Dylan Winkler. He says, great presentation with an exclamation point. I may have missed this, but are you transplanting or augmenting any of the roadside populations?
So many of the seeds that we collected for the augmentation did come from roadside populations. And we actually just recently, it wasn't included in this one because we just started doing it. We did another augmentation of the Flamingo Villas population just this, I guess, 2 Januaries ago. So it's about 1.5 years old. And those use seeds, yes, from the roadside populations. Most of that is due to, it's just they're easier to collect. There's a lot of them. When we apply for permits for collecting of wild plants, we have to say that we're not going to protect or collect over a certain percentage. And a lot of these gaps, or these plants occurring in these gaps, tend to be, they're flowering and they're doing well, but we don't want to take any of the propagules from them because they need that fire to go through there, which it has gone through finally. And so no, we have focused a lot of our collections along roadsides, mostly for ease, but also availability is what we were taking was less likely to harm the population.
Okay. Next question comes from Anna Dixon. She's curious, does this area have a large diversity of endemics, and how did you decide which species to use in your study? She does state that she missed the first part, so might be some details that you restate.
No, that's fine. So we selected this species mostly because we had some really good long demographic data that we felt that we understood a lot more about this species than we might for some of the other endemics that we don't necessarily study as in depth. But part of it also is that we got funding through U.S. Fish and Wildlife Service to do translocations of this species. And that is because it's one of the species that is, well, it's one of many species that is strongly encouraged that translocations may be in need in order to preserve populations. And with it only having one protected site, that was definitely a big push for why it should have why we translocated it or introduced it to a different site to kind of secure it across 2 different managed populations, and also to kind of provide a place for those unprotected genotypes that are just north of Flamingo Villas, have a place that they could be secured into the future through proper management.
Okay. There's a question from Jay Berger. He's curious, is there other research going on like this for other endangered plants in the area, and if so, which species?
Well, I'm going to encourage you to look at the Plant Ecology Program's website. Through the Archbold website, Dr. Eric Mangas has been working in the Florida scrub for about 30 years now. And a lot of his long-term demographic data sets are about 30 years old. At any given time, he has data on probably about 18 different he has detailed demographic data on probably about 18 different scrub species. And then we have maybe more coarse data on any of the 40 listed species, state listed species. So we do have lots of data, and we have used this data to do translocations of other species. One that comes to mind is the Avon Park Harbells, Curtularia avonensis. We've done 2 introductions of that species to a different protected site using genetic material from an unprotected site. And we used similar techniques to where we had long-term demographic data on wild populations. Based on that data, we kind of felt we knew what appropriate habitats we needed to look for, but we also had some questions that we wanted to answer. And so all of our translocations that we do in our program always have some sort of experimental component so that we can always learn more about the plant. Now, I didn't focus on some of our questions today. I wanted to more focus on just how these translocations compare to our wild populations. But we did learn quite a bit from both these translocations about this basic biology of this species.
Okay, I think we have one more question from Amartya over at Buck Island. He's curious, did you assume that soil moisture conditions in introduced areas are similar to that in existing wild populations? And then he comments, as in general, a lot of introductions fail because of underlying differences in rhizosphere water.
Yeah, so we didn't check soil moisture prior. We used the condition of, we looked at gap size and if it had other endemics present that we might expect to find at the Flamingo Villas population, that to us was suggesting that the gaps were probably functioning somewhat similar if they're able to support a similar suite of species. But no, we didn't look at soil moisture specifically. Like I said, we did provide supplemental irrigation, and specifically in the introduction, we used it as a treatment to where some received supplemental water and others didn't, and ultimately we saw no effect of that. Now again, we got really lucky with some really good rain years that year. In other years, you might see a treatment effect of providing water versus not providing water, but in this case, we did not see that.
There's actually another question coming from Buck Island from Greg this time. He says, you mentioned looking at management. Do you think the same management is likely to affect different gap specialists differently?
Yeah, I would think so. I mean, there's so much that goes into how these species, you know, every species requires its own management, but we can't manage for a single species. We have to manage for multiple species. And so that's why, especially in my intro, I mentioned that the fire range is so, or the fire return interval is so wide. It's perfect for some species and not for others. You know, we have other gap endemics that occur on white sands, in this case, Hypericum punicola and Eryngium punifolium. We see that if they don't have fire return interval, if they have fire return interval greater than 20 years, we tend to see them kind of bleep off the landscape. But in this case, for Dicerandra christmanti, some of those sites for the wild populations haven't been burned in decades. In fact, we're trying the process for the manuscript, trying to figure out when they did last burn, if we can even get a guess. But yet those plants are still holding on, and those gaps to some extent are still remaining, and they're recruiting. So that one has a much longer time to extirpation in terms of the lack of fire, as opposed to other endemics. And so that's why managing with a very diverse fire return interval and fire management plan can be beneficial for all species. I'd like to point out that for the Carter Creek population, U.S. Fish and Wildlife Service has been really great about getting fire back into that system. They had struggled for a couple of years, not because of their own efforts, but because it had just been so long unburned. It was just difficult to get fire on the ground and through the management units, but they've been trying for many years, and they're finally getting really good fires, to the point that when they had the recent fire that affected our introduction, it burned in a way that was very patchy, which is what we want in a Florida scrub ecosystem. We want patches to burn and other patches to not burn, because then you kind of end up with this mosaic of, you know, this species likes a really frequent fire return interval and it got it, so it's going to do really well in this gap. But this species likes something a little bit longer, so it's going to do really well in this gap that didn't burn this time. So you kind of end up with this really diverse management across the landscape that benefits all species.
Okay, and then I think officially, lastly, Nancy Bissett was curious because you showed 3 populations. If you'd be willing to explain more of like where they are, especially the unprotected populations, or. Yeah.
I have not personally been to those unprotected populations. Those were collected before I started working at Archbold. But I know that they went out and got, I believe they got the data from FNA, but it might have also just been local knowledge from previous surveys to basically go out and confirm if they were still there, and then getting landowner permission to be able to collect those propagules. I don't know if they've been checked since we were out there last, which would have been about 2011, to collect the information or to collect the material. But no, I have not personally been to those sites, but that's where we got the material from.
Okay, Dwayne Coltsman had another question. He's curious, how do you determine which plant clumps are different populations? So like, is it distance only, or are there genetic studies to sort of back up the space between these plant clumps?
Yeah, the definition of a population can be troublesome. I guess that's why I've been trying to move myself into referring them to as management treatments, because a population, we don't really know how isolated they may be. So when I talk about the Carter Creek ones, you know, those are somewhat a decent distance from the unprotected wild sites, but we don't know if there's gene flow going on between them. And so if there's no gene flow, are they isolated populations? But then within the Flamingo Villas site, you know, where we augmented, there were no plants, but there's wild plants growing on a road a couple 100 meters away. And so it's feasible that there might be gene flow going on that way. And if there's gene flow going through those, from our augmented plants to a roadside population, which then might be going to the wild gaps, you know, is the Flamingo Villas a population functioning as a population as a whole? Or so I need to get better about using its management treatments. Thank you for bringing that up. That some of them are going to be better connected than others, and that's basically, it's mostly on a spatial scale. But then also you have that there might be isolated pollinators, but then they might get a management treatment because it happens to occur in the same management unit. So there's a lot of different ways you can define a population. We haven't done genetics on this. It definitely would be an interesting question to see what the genetic diversity is and if they are truly isolated patches from each other, and that would definitely lead to support on why we kind of kept the genetics separate from the unprotected wild populations from the protected populations or protected sites, excuse me. But then that would also lend to do future augmentations or introductions. Do they need to maybe start to mix some of this genetic diversity, which is a whole other can of worms that is hard to decide.
Yeah, sometimes they find out in certain mammal species, there's like, you know, prolific reproduction occurring in a few individuals. Maybe they could find that with plants, but it'd be maybe a little tougher to do. That's all the questions I have listed. So I think that about sums it up. Thanks, Stephanie.
Thank you.